Hey guys, so welcome back again to another video. So in this video we are going to talk about another important molecule, biomolecule, which is the vitamin K2, right? So we are going to talk about how the biological systems synthesize this vitamin K2. So we are going to start our molecule with this particular reagent. Okay, we need actually two molecules, right? We need actually two, this one and this one. We need two of these molecules, but let's start with this one. So in the first step we are going to introduce this TPP to our original molecule. If you are not remember this TPP structure, this is the TPP structure. It's on here too. Let me introduce it again. So it's like this one. We have a nitrogen here, and we have a sulfur here, and we have a OPP group here, and here you have a R group. So you have two double bonds. This is basically the TPP, right? This is like a catalyst, right? This is like a catalyst. This has no effect, like it's not involving in the reaction, but it's behave like a catalyst. So when we are introducing the TPP to our original molecule, it will bind to this original molecule like this. So we have this structure, okay? So we have this structure, and now in the next step, what will happen is we are going to do the decarboxylation. Okay, here we have doing the decarboxylation. So decarboxylation happens, and then we are going to introduce a OH group instead of carboxyl group. So basically, this carboxylic group is going to be removed. Instead of this carboxylic group, we are going to introduce a OH group, right? We are going to introduce a OH group. So when this reaction happens, right? When this reaction happens, after this product form, the TPP group will be removed. Okay? So in here also, the TPP is removed. TPP is removed. Sometimes you may see that textbook content like the TPP removing step including in the here, but the TPP removing process is including in here. It's fine, actually it's fine, because like TPP has no overall effect in your mechanism, so that's like it's fine to like remove it in here or here, because like when it comes to this particular place, the function of TPP is over, right? Basically the function of TPP is like to as a protecting group, right? It's protect this carbonyl, it's protecting these other groups to avoid reactions. So here we have this part. Now the TPP is removed. We have a nucleophilic part. We have a nucleophilic group in this place. We have a nucleophilic group in this place. So TPP has introduced a nucleophilic in this place. Now what happens? This nucleophilic group, let me take another color. Okay, we will take blue color. Okay, so this nucleophilic group will go and attack to this particular position. Okay, so I hope you are okay. Now when this attacking happen, this bond will be broken and form over here. Now we will have this kind of molecule here, you can see. So this same thing happened, right? So when this attack happens here, when this attack occurs, this hydroxyl group convert into a carbonyl group again, right? So you can see when this attack happen, this carbon is needing another electron, so oxygen is giving another electrons to it, so hydrogen will be removed. So we have this carbonyl group. Now in the next step, now we have this complicated molecule. Now in the next step, what happens is, right? So two steps, like two scenarios will be happen. The first one is the releasing of this H2O group. So this OH group here, right? This OH group will be removed alongside with the hydrogen on here. Right? So when this happen, this bond will be formed back again. So this will break to form a double bond here. And nextly, this group, right? This whole group will be broken, and then it will be removed as like this. This is not oxygen, right? Again, like some people are just confusing with this, like when we are taking the notes likewise, so then we are going to look at the notes in later days, and we will think like we are missing the oxygen here. No, it's right. This is a CH2 group. Okay, this is not oxygen. So I hope you're okay up to now. So hang on with me. Now we have, when we are doing these two processes, we have this particular molecule. Okay, we have this molecule. I just forgot to mention the name about this molecule. This is another reagent. Like, I mean, I have told you that we are going to need two molecules. We are going to need two biomolecules. The first one is this one, and the second one is this one. This is like isochromic acid. Isochromic acid. Okay, right. Now let's move on. So now we are here, and then the next step. Okay, in the next step, you can see what happens is we are going to introduce enzymes to this carboxyl group. Okay, here this carboxyl group is going to be converted into a enzyme group. So we are basically going to introduce this type of an ATP. ATP is needed to give the energy, right? So we are going to obtain this molecule with this, and then what happens is a basic Claisen condensation or Claisen reaction. Claisen reaction happens. I hope you know about the Claisen reactions. Again, these Claisen reactions are general reactions in biosynthesis pathways. So if you are not familiar with these Claisen reactions and aldol reactions, I highly recommend you to go back to our previous videos and refresh those memories. These are very important. Okay, now when the Claisen reaction happens, you know, like this place, you know, basically there are hydrogens. So with a proper biological base, we can remove this hydrogen and make an electron lone pair in this particular position. This electron lone pair has nucleophilic characteristics. So now this lone pair will go and attack to this carbonyl group, which has electrophilic characteristics, right? So with the Claisen mechanism, you know, when the Claisen mechanism happens, this OH group which is attached to this carbon is going to be removed, right? That's the Claisen reaction. That's the Claisen mechanism. So when that happens, you can see here this bond over here, and this carbon here, look at this carbon is going to be this place, and a new bond will be formed because of this attack between these two. So now this group is going to be moved over here. So that's what I have drawn on this place, the final product of our Claisen mechanism. Now we have this product on here, and then what happens is, so this is basically the same molecule, right? Basically the same molecule. Now hydrolysis will be happen. Hydrolysis will be happen, and the forming of double bonds. So you can see these carbonyl groups here. These carbonyl groups is being converted into hydroxyl group, and two double bonds is going to be formed. So here we have the hydroxyl group, hydrolysis part, and then in the next step, we are going to introduce again our initial that polymer molecule. If you remember this one, yeah, you have this one. Sorry, not H here. This one, okay, this molecule, right? So this carboxyl group will be replaced with this part. So there are these phosphate groups. These phosphate groups will be removed alongside. So here, if I number them, 1, 2, 3, 4. Yeah, 1, 2, 3, 4. Likewise. So this carbon is the carbon. This carbon is the carbon which is going to be form the bond between this our ring. Now I hope you are okay with it. In the next step, okay. So in the next step, so this just forget it. In the next step, what happens is we are going to introduce another reagent, not a reagent basically. What we want to do is the oxidation. So these OH groups has to be converted back into the carbonyl groups like this. Okay, carbonyl groups. This, so we can, if you remember, like how we can remove these hydrogens, it's basically NAD plus. NAD plus is the one. NAD plus is the coenzyme which is being used to remove this hydrogen and form the oxygen carbon double bond. And also you can see there's a methyl group is being attached to here. So I hope you know how to attach a methyl group, right? This basically a methylation. It's a carbon methylation. So basically there are SAM. SAM is the molecule which is going to do the methylation part. So here we have our final molecule. This is the vitamin K2. So it's a little bit like there are some steps, right? This is not a simple step mechanism, but if you look closely, like if you understand the mechanism, you will understand clearly that only difference is the molecules, right? These molecules are a little bit complicated. That's the only thing. The mechanisms are just the same. You can see introducing, like in first step we have the decarboxylation, basically the decarboxylation. So here this part will be removed, and instead of that we have OH2. So like basically this oxygen is going to be taken a proton to become this hydroxyl group. Likewise, these are simple reactions, right? These are simple reactions. Only difference is these molecules. The molecules are changing. Molecules are changing. These electron densities, these are changing. That's how this mechanism get a little bit tricky. So understand the structure, understand the core concept of a mechanism, then you can easily do answer the question regarding this mechanism. So basically, like I have told you now previous video, just understand the general reactions, right? Understand the decarboxylation's mechanism, understand like how the dehydration happens. This is basically fine, right? You know, this is a good living group. So like otherwise, how's the hydrolysis happening, how's the oxidation happening, what is the NAD plus, how this NAD plus behave with other molecules, how the this methylation happens, how that happens, what's the mechanism. Understand these core reactions, then you can easily follow up these biosynthesis pathways. These are not hard work, these are not complicated work. When you are understanding the basic concept, you will feel like it's completely fine. So I hope you get something from this. In our next video we will look more into these biosynthesis pathways.